First announcement for today is Student Research Symposium tomorrow over in Smith Center in the ballroom. It's usually a lot of fun, cool posters on what people have been doing in various different labs in various different departments all over campus. Again, usually a lot of fun. I think the poster session is from 11 to 1 officially. So anybody have a poster over there? Yes? No? Not going to admit it? Good. So hopefully we'll see you all over there tomorrow. You'll need a study break anyway, right? Because what's happening on Wednesday? Midterm. Good. When are we going to be here? 9 o'clock. How many seats are going to be empty? None? Okay, good. So we're all on the same page as far as that's concerned. And yes, we do have a couple of clicker questions today. But as soon as those are done, you may leave. Well, try not to be too obnoxious about it. So today's starting question is, a calimovirus, probably infects cucumbers, tomato, caterpillars, cauliflower, or humans. The original thing that's infected. So, like cucumber mosaic virus infects things. Pardon? Yeah. One more person voted. Good. Any more? Yes? No? So in theory, you don't get points for coming, right? In practice, yeah, it looks like you do get points for coming. So yeah, it's this cauliflower mosaic virus. The TWIV this week was all about cauliflower mosaic virus. That actually is a new families of cauliflower mosaic virus that infecting grapevines and all kinds of other things. So the important thing here is that's the name of the original species and phenotype that it gives, but something like cucumber mosaic virus can infect all kinds of different species. And so the point here is, yes, that's what this particular one is named, but it could be infecting a bunch of other things, although highly unlikely to be these guys here. So that being said, let's quickly select the answer and move on. Second question. Unlike most capsid protein structures we've discussed so far, the TMV capsid protein is made of mostly alpha helix, 1 beta barrel, 2 beta barrels, mixture of alpha helices and beta sheets, or a 10-stranded beta tunnel. $30. $20. $10. 5. Okay, we've got a mixture here. Lots of different ideas. The correct answer is A. It's mostly alpha helical. What? Unlike most of the capsid protein structures that we've talked about so far, most of which are beta barrels. In terms of most of the RNA viruses we've talked about so far, what are most of them? Single beta barrel. If you think about the poliovirus capsid, it's mostly this single beta barrel. And a 10-stranded beta tunnel. What the heck was that about?
That was like the injector.
The injector thing, right, from which one? Not potoviruses, no. But yes, a bacterial virus. Phi-X174, yes, that is correct. So that's it for clickers for today. Those of you who have to leave, adios. Best of luck studying. Any questions, please email me. I will do my best to answer them until about 10 o'clock tonight. After that, no guarantees that I will actually get around to answering your questions. So this exam is on RNA and RNA-only viruses. So things like polio on the positive strand, and then the various different negative strand viruses. Just like I do for all exams, I basically go through the lectures, and there should be about the same number of questions per lecture, and you can do the math. How many lectures do we have? 7, 50 questions. So what does that leave you with? About 7 per lecture. So that's how you should think about it. I don't have 7 slides per lecture. There's no guarantee whatsoever that the slides I present today will have anything to do at all with the exam. But these are the things that I think either did a bad job of explaining the first time around, or I think deserve some more explanation. As far as polio, it's not so much the disease, but really about vaccines. And we talk a little bit about vaccines, actually quite a bit, this particular section of the course. So it wouldn't surprise me if there are some vaccine questions that are on the exam. But basically the idea here is there are really 2 different kinds of vaccines in general. We either have inactivated vaccines, which is where you purify a whole bunch of virions and inactivate them somehow so that they can no longer replicate. Or you have the attenuated versions, which are those which are actual infectious viruses that don't cause disease. And the original vaccines that were developed were in fact these attenuated vaccines, not for polio. First polio vaccine was in fact this inactivated one. But that's the basic idea in terms of these vaccines. Attenuated originally was in passaging the viruses through individual animals. Now it's much more done in cell culture. We talked about some other vaccines, which are now the recombinant vaccines, where you're mixing and matching different kinds of things here. But those we'll come back to a little bit later on. As far as polio eradication, we're getting really close. A lot of that has to do with this oral polio vaccine that we have here. Why is that? It's easy to administer, yes. Don't have to give shots like you have to over here. What's another reason? It can spread. You don't actually have to necessarily be immunizing everyone directly here with the drops. Whatever comes out the other end of this tube can also be used for vaccination per se. What's the problem with this? Can actually cause disease in some cases because attenuation has just changed a couple of things in the genome, and so those on a random basis can change back. So a lot of the polio cases in places where we've got good vaccination actually come from this oral polio vaccine, and what that means is if we really want to make sure that we're done with polio or nobody makes polio from scratch as they can, then we're certainly going to need some of this inactivated polio vaccine as well. But that's the idea. So vaccines aside, disease aside, what are some of the really fascinating aspects of polio and the picoRNA viruses in general? It's that they do things kind of like T7 and FIX174. They inject their genome directly through the plasma membrane, again, in a manner very similar to the bacteriophages. The process here, though, as well, is just like we've had and talked about with pretty much all of the other receptor binding and entry. It's a conformational change. And here in particular, it's conformational change of, we use the VP1 protein and the VP4 protein that make this channel to go through here. One thing that I think I didn't mention the last time around really has to do with the poliovirus receptor. Got a couple of immunoglobulin-like domains, again, like many of the cellular receptors. Here, however, instead of binding at the very outside of the virion, you'd sort of expect it to bind down here. It actually binds up here. And this binding and conformational change that happens here, particularly having to do with what's called the canyon here. You may have seen this in some of the previous exams. This is actually a really good target for antiviral drugs. If you block this here, you can't have the conformational change. You can't get the genome, which is injected inside the cell. What does that genome look like? Hopefully polyproteins are now something that are near and dear to you, as opposed to what the heck are these things before? So polyproteins, very common in these positive strand RNA viruses. In the case of polio, the genome actually has one of these proteins which has gotten chopped off and attached to one of the ends. But all of the proteins here are encoded in one ginormous open reading frame. They're all made as one protein. And then, of course, they need to get chopped up into smaller pieces. Yeah?
So with the, you just said that VPG protein?
Yeah.
So since there's a segment of it, you just said that it gets chopped out?
Right. We'll talk a little bit more about genome replication in just a second. So what gets injected through that conformational change in the pore that happens is VPG connected to the rest of the genome. But you can't reuse that VPG, and for more genome replication, you've got to get this VPG cleaved out of that polyprotein in order to use it for the next step. Yeah?
Is it considered a 3B protein until you're still added and then it becomes a VPG protein?
So, okay, yeah, nomenclature-wise, what does Stedman going to expect you to know in terms of all of these letters and numbers for the exam? In a case like this, I would say VPG. And VPG and 3B are used really interchangeably with each other. But since I've used VPG way more, that's what I would be thinking about. So things like that. As far as numbers that I would think about in terms of polio, I would say 3C and 3D, particularly 3C. Why 3C? That's the protease, exactly. That's the one which is doing most of the job in terms of chopping everything up. I wouldn't remember all of the other ones, 2C, 2A, 2B, etc. To be or not to be, that's so yeah, getting back to VPG and where VPG comes into this whole process. Again, the genome, as it's released inside the cell, has VPG attached to one end, and the extreme 5' end of the sequence has 2 U's on it. The 3' end, of course, has a poly A tail. This is its normal process that you would get the translation happening here. Where does that poly A tail come from? How's it made? It's not made by stuttering. It's part of the genome. It's encoded in the genome. And this is a difference here between some of the, particularly the negative strand viruses. Here, that poly A tail is part of the genome. So this is that structure. Again, you've got a poly A tail here. You've got VPG bound here. You have a number of cellular proteins, particularly this poly C binding protein here. not surprisingly, binds to a whole set of C residues, but more importantly, a secondary structure present here, the 5' end of the genome, as well as your polymerase structure. This polymerase structure basically recognizes this free VPG, because it's just been made cleavage-wise, and then puts these 2 U residues on it. So these 2 U's then serve as primers for the RNA-dependent RNA polymerase, also known as 3D, to make a negative strand of your genome. That negative strand comes all the way over here to the other end. At the other end, you've got 2 U residues. So those become 2 A's in your negative strand. VPG gets 2 U's added to it, and then extends the rest of the genome. Where do those 2 U's come from? They actually come from this guy right here in the middle. Again, a particular secondary structure which associates with these polymerases and VPG and gives you a couple of U residues. So it's really kind of a convoluted mechanism how this happens. So this is your genome. VPG is produced. First couple of U's happen here. Then they bind to this end. Provides the primer for negative strand synthesis. Then we have VPG with a couple of U's on it, which serves as the primer for positive strand synthesis. Yes, no, maybe, huh? Okay, what does this? Again, it's 3D. It's a DNA polymerase like fold. So basically, it needs a primer. It needs a template. Primer here is provided by VPG, and the template is just your genome. This RNA-dependent RNA polymerase, just like all RNA-dependent RNA polymerases, doesn't do what? Editing proofreading. So this structure here, yes, looks like a DNA polymerase. All of the proofreading that happens in DNA polymerases is due to an extra domain which is hanging off the end of those DNA polymerases which do proofreading. These guys don't. But otherwise, the structure is extremely similar. Questions on picoRNA viruses, polio? Yeah. So coronavirus has a different protein which helps for proofreading. So we'll get there. Although it's great, we can skip it next time, right? So we get there now. Okay, so now I put a membrane on these picoRNA viruses, and we call them Flaviviruses, and we get lots of nasty diseases. That's, again, molecularly speaking, that's about the only difference that you have between these guys. How you get disease here, however, has to do with most of these being vector-borne diseases, yellow fever, dengue, and West Nile. Hep C is the big exception to that because it's a human-to-human blood transmission that happens. Dengue, probably the most common viral disease on the planet right now, with the possible exception of influenza. And we can worry about numbers a little bit later on, but definitely expanding. This is one of the emerging diseases that is causing more and more of a problem, mostly through presence of these mosquitoes, and numbers are going way, way up here. Partly also because we don't have a good vaccine, although it's getting very close right now. We've got a couple of vaccines that have gone through trials and do seem to be really helping. What do these virions actually look like? Again, they're basically picoRNA viruses with a membrane around the outside, and then, of course, membrane proteins, which are sticking into that membrane. But those membrane proteins are so close to each other that this almost looks like a naked particle. There's actually very little membrane showing when you look at these virions under the microscope. Again, the genome is extremely similar, except here it has a cap at one end. What does that presence of a cap mean? There's something which is putting it on there. We've had one example of where caps get stolen, but in all other cases, what happens? It's made by some of these viral proteins, and since I say some of them, I'm not going to ask you which one. But cap is made here. Then we've got our structural proteins here at the 5' end of the genome, the non-structural proteins over here, also made as a polyprotein, also cleaved up by viral proteases. Big difference here is that we also have cellular proteases that are involved in cleaving up that polyprotein. Some of them are due to the fact that these are membrane proteins, signal peptidases. This is what cleaves normal membrane proteins, cellular membrane proteins as well. And then one extra protease, this is a cellular protease, the furin protease, which seems to be mostly involved in sort of the very last steps of what we call virion maturation, making sure it's actually infectious. And this protease, this cellular protease, is also the same one which is being used in influenza to chop HA0 into HA1 and HA2. So really a quite common occurrence here. But otherwise, extremely similar. Which of these proteins is the one, actually there are two of them, that are the targets of these new drugs that actually do seem to really work very well against hepatitis C? So the protease and the polymerase. So NS3 and NS5. Questions on the flaviviruses? Yeah?
Back to one more slide. So in the book, the PRM thing,
Yeah.
so it gets cleaved into the M protein and it loses PM,
Mm-hmm.
or PR. What exactly is PR again? What does it stand for?
So PR is just pro. So it's the pro-M protein or the precursor to M protein. A lot of people will talk about PR for precursor or pro. It just depends on how you look at it. When we talk about HIV in the dim, dark, distant future, i.e. Friday, we'll talk about some of the PR proteins there as well.
You said NS3 and NS5 were the protease and
The RNA-dependent RNA polymerase. And those are the drugs that do seem to really cure Hep C if you have an insurance company that's willing to spend $100,000. Okay, other questions? Sorry, any other questions on the flaviviruses before we move on? Okay, now finally, the coronaviruses. You're asking about back up in the back there. So these are also still positive strand RNA viruses. Really big genomes, but they have some proteins that are encoded in them that seem to be important for replication in terms of the proofreading, correcting some of the things that happen with them. But before that, the order that I put these in, the way that we talked about them, has to do with where some of these coronaviruses came from. It's the one that we cared about at least till a couple of years ago the most. SARS, severe acute respiratory syndrome virus. If you look at the sequence of this virus, it's extremely similar to the sequence of the civet cat virus, so that's why people thought originally that humans were probably infected from civet cats, but that seems a lot less likely because if you look at the diversity of other coronaviruses, most of them are in what? Bat, 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 all the way over here. And that's how you have one indication of a reservoir species, that you've got huge amounts of diversity in the viruses in something that you think is going to be where that virus is normally circulating. What other thing do you usually expect in a reservoir species? Not a lot of symptoms, not a lot of disease, which also seems to be the case for most of these bad coronaviruses as well. So, not much disease, lots of sequence diversity in them. Probably that's where the reservoir is. Okay, what are these genomes? They're ridiculously enormous, at least for positive strand RNA viruses, up to a little bit over 30,000 bases in length. And probably because of that length, they need to have some of these repair proteins, particularly this one here, NS14, which is the one which seems to be important for that basically proofreading-like activity. Usually if you talk about proofreading activity for a polymerase, it's going to be a separate domain. It's going to be the polymerase itself that does that. It's a separate protein here that is helping out with that. That protein, however, is made, as true for most of these positive strand RNA viruses, part of a polyprotein. The polyprotein for the coronaviruses is different because it's present in two different open reading frames. What that means is when this is being translated, a few times the ribosome, the amino acid polymerase, will shift reading frames and continue, still making one long polypeptide chain where it makes this polypeptide connected to this one through, again, a peptide bond. Once again, you need various different proteases to chop all of this apart. Each of those are the important ones in terms of splitting out all of these different proteins. We haven't talked very much about these guys, so I wouldn't worry about memorizing all of these names. The big difference, of course, as far as all of these coronaviruses has to do with the structural proteins. Unlike Flavis and Picos, they're all encoded down here at the 3' end rather than at the 5' end of the genome. And why is that? That's because there are lots of subgenomic RNAs, subgenomic messenger RNAs that are made to make these structural proteins that are important for the structure of each of these coronavirus virions. The way this works is through recombination, which is probably the other way that coronaviruses can get away with having these ridiculously large genomes, than switching back and forth between templates. So how does that happen? You have an RNA-dependent RNA polymerase, starts out here at the 3' end of the genome, synthesizes happily away until it hits one of these TRS sequences. The sequence here is not important, at least not as far as the exam is concerned. Hits one of these sequences and then can either keep polymerizing or jump to one of the other ones, and in particular this TRS sequence right here, hidden behind this guy, where you end up copying the very 5' end of your genome. The very 5' end of your genome is really important for getting good translation. So any of these pieces here, you jump over, you end up with this leader sequence. These are now all, right here, negative strands. So these are not going to be coding protein. They have to be copied yet again by the RNA-dependent RNA polymerase to make these subgenomic RNAs. Because all these subgenomic RNAs have different lengths, the one RNA that you have the most of is going to be the one that's coded for at the very 3' end of your genome, and that's, of course, the protein that you need most of in your virion. And then less and less of these subgenomic messenger RNAs relative here. These are also called nested subgenomic RNAs because this one fits inside that one, fits inside this one, et cetera, et cetera. And these viruses are also called the nidoviruses for the nests that are on the inside here. The other coronavirus that people are caring about a lot is MERS. Again, last 5 years or so. Clearly been in camels a lot longer than that. But what's a little odd is that these guys have an identical sequence to the humans. What does that mean in terms of camels potentially being a reservoir species? Pardon? Well, it's quite possible that these guys are infecting the humans. But what do we talk about in the bats? What do you see in the bats that's different? Huge amounts of diversity, whereas here you don't see a massive amount of diversity. The camels don't seem to be sick, so that's one aspect of it. But you don't see a huge amount of diversity there. That's partly why people are looking a little further in terms of trying to find out where maybe a different reservoir species is relative to the camels. So questions about any of those positive strand RNA viruses before we shift over to the negative side? Yeah?
I was just wondering, if you get MERS,
Yeah.
I thought you had said that you are more likely to die than if you got killed. Like, it kills more people?
Yes, that is That is correct. If you are diagnosed with MERS, and that's a big diagnosis thing, separation there. Diagnosed with MERS, usually you're really, really sick, and that leads to pretty high mortality rate in terms of diagnosis per mortality. Dengue, on the other hand, there's a lot more diagnoses of dengue, and fewer people end up dying. But again, it's this nominator-denominator issue in terms of the actual numbers there. So yeah, there probably are many more cases of MERS that people just don't notice or don't particularly diagnose. So that's one of the big problems with a lot of this epidemiology and trying to follow up these things. Okay, negative strands. So first and immediately obvious, hopefully, here is just thinking about virions. If you think about virions, negative strand RNA viruses are bringing the whole kitchen sink with them. So it's not just one nice positive strand RNA, that's all you need. Once that gets inside the cell, it gets translated, everything's happy. Here, no. You've got to have your own RNA-dependent RNA polymerase, and very often lots of other proteins which are present there as well. And that's the main reason that I picked this image here. Again, from the viral zone, nice place to go to review any of the individual viruses. Nudge, nudge, wink, wink. So here we've got the viral genome, which is packaged with nucleoprotein. Also, you have your polymerase and accessory polymerase proteins. All of these together packaged inside the virion. This virion, in the case of the Paramyxoviruses, like measles, has a hemagglutinin and a fusion protein, two separate proteins, a matrix protein, and then other proteins as well. But the main thing here is we've got lots and lots of proteins all present in the virion. Measles, of course, is going way up, and this was just literally from this year. I don't have the up-to-date numbers since our last lecture. I think it's gotten over 200 now. Most of that is having to do with people who are not vaccinated, either by choice or because they can't be vaccinated. All of these cases have been imported into the US. So none of them are, we don't have a circulating measles here in the US because of this absolutely wonderful vaccine, exactly, which everyone should be getting. So that protects you against replication of these kinds of viruses. These are the Paramyxoviruses, the rhabdoviruses, and the filoviruses have one genome segment with a bunch of open reading frames in it. But of course, relative to all the viruses we've talked about so far today, they start at 3' and go to 5' because they're negative strand. This has the great advantage that the RNA-dependent RNA polymerase can start here and make messenger RNAs immediately, as opposed to having to go through that whole convoluted process we saw with the coronaviruses last time. You know, making part of it, recombining, making all these subgenomics, which then end up getting recopied into your messenger RNAs. Here, you're ready to go as soon as you get inside the cell because you've got your polymerase already present, sitting here at the 3' end of the genome, ready to go. Makes a messenger RNA for the nucleocapsid protein, gets to one of these intervening sequences, and now we get stuttering, exactly, which gives us a poly A tail at the end of this particular messenger RNA. After we get through that stuttering, there are two possibilities. Either the polymerase falls off, goes back to the beginning, and does it again, or it stays associated with the genome and starts again, makes another piece here, stutters at the end, makes a piece, stutters, etc. All the way through down to the very far end of the genome. And this brings me to something that I forgot to mention. We did this lecture before. How do we know that that's the case? Well, the best evidence is experimentally if you make a mutation or a deletion in this particular open reading frame, just one in the middle. In this case, it's the M protein, but it's not really critical exactly which one it is. Then, if you get the polymerase to stop here, none of the rest of the genes down here get made. And so one of the theories about how each of these open reading frames was being made was that the polymerase could come in either here, or here, or here, or here, or here, etc. So here, the fact you make a mutation in one gene stops the production of all of the genes downstream of that. That means that we have this so-called single entry model, or the polymerase doesn't dissociate from the template as it's making the messenger RNAs. In Paramyxoviruses, rhabdoviruses, and the filoviruses, you have RNA editing. I don't like the term, but that's what everybody calls it, where you have some basically stuttering that happens not just at poly A tails, but also in coding sequences. And in that coding sequence, you can have a change of everything in your protein, which is going to be downstream or C-terminal of wherever that change happened to be. So in the Paramyxoviruses, mostly you get the P protein, and you get some of this V protein due to this RNA editing. This open reading frame, I should say this piece of genome, has multiple open reading frames in it. So if you start at a different place, and that's what this is shown down here, which is not in the same frame, not the same multiple of 3 as where you have this P protein, you can end up with a completely different protein. And that's the C protein that you have in the Paramyxoviruses. So 2 different ways that you can get different proteins out of this. One is stuttering inside the open reading frame. The other one is using an alternative start codon. Okay, questions about these guys? Now our friends, the filoviruses. Liberia officially declared Ebola-free. Anybody hear that this morning? Very good news. Amazing how quickly that happened. I think everyone's kind of surprised, but very happy. So seems to be mostly through public health and communication. Really, a lot of this had to do with communication, not so much in terms of, great medicine. Really had to do with the communication going through there. So these guys, one of the things that unfortunately is not on here, these virions are humongous. They're actually some of the largest virions that we'll talk about in this class until we talk about the giant viruses right at the end. These guys are very large, and that's actually partly why, probably anyway, it's hard to get transmission for a lot of these filoviruses. The virions are so big that you have to have direct contact, bodily fluids, etc. They don't go nicely into aerosols, unlike things like measles, which are really good again transmitted through aerosols. So again, negative strand, non-segmented genome. It's got its polymerase hanging down here at one end, glycoproteins on the outside. Major difference between these two is this VP30 protein, which is a translational regulator, and this secreted glycoprotein versus the main glycoprotein. The secreted glycoprotein is actually made much more than the non-secreted glycoprotein. And so this is, again, a bit of a difference between this and then the paramyxos and rhabdos, which we have down here at the bottom. One of the things that I didn't mention before, at least didn't emphasize, these all have 5' caps. What does that mean? Just like we have with the positive strand RNA viruses. They have their own enzymes that do this. Same thing is true for the paramyxos and the rhabdos. They also make caps through their polymerases, the L proteins. Here, very similar to what you have in the paramyxos and the rhabdoviruses, starting, stopping, the poly A tail formation, also through stuttering. Yeah?
That secreted glycoprotein, that's just
So that's the theory. The theory is that that's floating around in the blood and blocking up the immune system, either overstimulating the immune system, which is part of the problem, in fact, with the filovirus diseases, or not allowing the immune system to finally find these virions. So blocking that process. Yeah?
Another question with that. So that's done with like the RNA editing process as well, with like the stuttering, coughing?
Yeah, exactly. And although in this case, the non-stuttering case is your secreted glycoprotein, and then stuttering gives you the glycoprotein. And so this is a good explanation for why you have way more of the secreted glycoprotein than you have of the membrane-bound one, because stuttering doesn't happen all the time. Okay, disease, again, most of Ebola virus and Marburg viruses, very small numbers, very contained until about 2005. And then, of course, this year, and last year for that matter, really got rather out of control. But what's impressive here, again, is Liberia's gone from the most deaths to 0 cases now. And literally about 11,000 deaths compared to the 500 that we had before. Yeah?
How can they have more deaths than confirmed cases?
Yes, so that's a great question, and that was one of the questions that I asked myself as well when I looked at this. These are all going to have a little bit of floppy room to them, and these may also be deaths that happened in Ebola treatment centers when they weren't able to even do any kinds of diagnosis on them. If you have a chance, maybe after the midterm, to watch the PBS broadcast, they talk a lot about this, and they were really frustrated, particularly at the height of the disease in October, where you can't do any kind of post-mortems or anything. You've got to go to the next person, because that's what you got to do. You got to get the dead out to move more people in. And I think a lot of that comes from this. Particularly, again, in Liberia, where they had these really massive numbers. Everyone's worried, but no reason to be worried in the U.S. here. So questions about Ebola and filoviruses, etc. Something to be much more concerned about in the U.S. are these guys, the orthomyxoviruses, also known as flu. Again, negative strand RNA viruses bringing the whole kitchen sink along with the virion. They've got hemagglutinin, again, classic receptor binding protein, neuraminidase, which chops off the receptor, probably critical for getting release of the virions. This is the target of those drugs which are still working, neuraminidase inhibitors. So clearly that release process does seem to be important for viral disease. These are segmented genomes. So you've got, in the case of influenza A, 8 different little mini chromosomes. Again, I like to think of those with the RNA polymerase bound to the end of each of these individual genome segments, again, or chromosomes. How do these guys make messenger RNA? Again, the big difference for these guys is they steal caps. And they steal caps where the caps are, which is in the nucleus. So you've got to get this whole process, each of those individual chromosomes bound with all of their nucleocapsid proteins, get them inside the nucleus, regular nuclear transport mechanisms. Once they're there, they steal the caps. There are extra caps there because some of the non-structural proteins will block making of tails and export of RNAs. So there's an accumulation of cellular messenger RNAs with yummy caps on them to get picked up there. Once these caps are cleaved off by the polymerase molecules, these now will make a messenger RNA together with stuttering down here at the end. Cap comes from the cellular RNA. Poly A tail comes from stuttering. Now you have viral messenger RNA. That viral messenger RNA can make the proteins, which then get imported back into the nucleus. Those combine with the genomes. Genomes, in this case, and all of the other negative strand RNA viruses, how does the polymerase know what to package as a genome and what to leave as a messenger RNA? Concentration of the N or nucleocapsid protein. So high protein, make genome, package that genome, get it out. Low N protein, keep making enough messenger RNA until you have enough of those proteins in order to put the virion together and have those go out. Yeah?
What is that ghost PB1 trying to represent?
What is the ghost PB1 trying to represent here? In this case, it's saying that this protein is bound. I don't have the slide here, but if we go back here, you'll notice that the PB1, PB2, and PA are all bound at the 5' and 3' ends of the genome. And so this is supposed to represent that it's bound here. Also, that this U tract is not all the way down at the 5' end of the genome. So it belongs. This piece here is never copied unless it's the genome. And so that's what that's supposed to be representing there, is you get to this point and you stop. And that's probably how you're getting part of that difference. When you've got the N protein bound, that's going to be binding up, including some of these sequences, so you're not going to be stuttering here. So yeah, so this is shown as a nice linear piece here, but these guys are all bound together as loops. And you can even see them again in that wonderful micrograph that I showed at the beginning, where you can see literally each of those individual segments present inside the virion. Why should a lot more people care about flu and flu virus? Oh yeah, sorry.
Each one of those chromosomes encodes for like one protein.
Right, with two exceptions, which are the incompletely spliced versions here. So you end up with 10 proteins. So 8 chromosomes, 10 proteins for influenza A. The other molecular questions here. They want to talk about this year. This is this year in the US. Most of infection peaks really right at the change of the years. Literally here, week 1 of 2015, highest amount. Vast majority of these guys are influenza A. This is influenza A that they didn't check. Here, influenza A of the H3 type. H stands for hemagglutinin, N stands for neuraminidase. So that's how you can tell which ones they are. And H1N1, 2009, the swine flu, air quotes there for those people listening online, which really basically almost none of this year. So really pretty much completely gone. A lot less of influenza B. The fact that influenza B is coming up later this year, that's not particularly normal. That's going to be influenza B all over the place. But important here is that spikes right here, end of the year, beginning of the year, and mostly influenza A. In this particular year, the H3 vaccine wasn't a really good match, so we actually had a lot more of the H3 than of the H1. Normal influenza mortality in the US, about 36,000 times more than Ebola deaths ever. So much bigger deal. Most people older, and then many, many, many deaths worldwide. And a lot of times this again has to do with whenever you have an influenza mortality, actually a lot like MERS, there's usually a different disease as well present there. And so these are comorbidities. They might or might not have died if they had just that other one, but the two diseases together are what really leads to mortality. And a lot of these people, of course, are the older people who are there. We didn't talk about any of the pandemic flu stuff, which is all the notes at the end. That's not going to be on the exam. Yeah?
Back to like the H1N1 or just the H. So H3.
Yeah.
So do they name it H3, that specific type, or is it
Okay, yeah. So here, the way that, so back up a little bit. How do you type these different kinds of influenza? Basically, what you're looking for is antibodies, and what do they recognize? And so you've got antibodies that are going to interact with H1. You've got antibodies that interact with H3. And so that's the way you can tell, for instance, here, that these guys are all H3. They've actually been tested with that one particular antibody. Here, for the orange ones that we don't actually see this year, those can be, they have to be antibodies to both the hemagglutinin and the neuraminidase, or in many cases it's a sequencing. They use PCR, reverse transcriptase PCR, in order to do that. But they do a lot less of the reverse transcriptase PCR than you're going to do with antibodies. Antibodies are a very fast test to do. Unfortunately, I don't know if anyone here has done that testing. I never have. But there are basically tests where you take a sputum sample, put it onto a card, and then it'll light up with one of those particular ones which is in there. And usually it's the hemagglutinin that's being used. So yes, be much more concerned about flu than you are about Ebola. One person is not going to be able to do this, so all of you can go out and evangelize for me. Continue to do this. So, something completely different, but again, my friends at OSU, I haven't gotten death threats yet, but they obviously don't look at my lectures on YouTube. Different plant viruses, and again, mostly here when we're talking about plant viruses, we're talking about cucumber mosaic virus and tobacco mosaic virus. Two of the literally thousands of different viruses that you have that infect plants. But many of these viruses, like cucumber mosaic virus, have this really, I don't know what the best adjective is here, strange way of doing infections, and that's multiple capsids for your segmented genome, each of which has one, or in some cases, multiple RNAs in them. And so this case here, which is, again, I think I did a not very good job of explaining, cucumber mosaic virus has one capsid that has RNA1 in it, one capsid that has RNA2 in it, and then one that has both 3 and 4 in it. And the way that seems to happen, again, these are nice, pretty T equals 3 quasi-equivalent icosahedral capsids with what kind of capsid protein? Single beta barrel capsid protein that fit together really nice into these quasi-equivalent T equals 3 capsids. Each of those capsids can package a little over 3,000 nucleotides. So that's how you get this specific packaging. So unlike the case in influenza, where we really have no clue how you've got these 8 different segments that are all getting mixed and matched and put together, which is partly how you end up with things like swine flu in 2009. Here, just the size of the virion controls how big the RNA is that can get in. So you can't have more than one of these RNA1s. You can't have more than one RNA2. But you can fit in RNA3 and RNA4 together, because those together are a little over 3,000 nucleotides. So it's the size of the capsid which controls what things are actually getting packaged inside them. But to get a productive infection, you've got to get all 3 of these different capsids infecting one cell at one time, and that seems to be mostly because plant cells are really tough. And so they've got this nice, tough outside. How do you get viruses in the inside? You either have some poor undergraduate who's taking sandpaper and putting it on the leaf, which is literally how you do some of these things. Scratch up the leaf and then put your concentrated virus prep on it, or some kind of insect which is chewing on the leaf. In the case of cucumber mosaic virus, almost always aphids that are doing this. And to get good infection, you have to have then clearly very large numbers of viruses to be there. Each of these RNA segments has a really interesting structure at the 3' end of its genome. It looks so much like a transfer RNA that amino-acyl tRNA synthetases will bind to it and even put an amino acid at the end, which provides detection from the nasty exonucleases, which would just love to chew up your RNAs, which are present inside the cell. These guys also all have caps at the end of the genome, which means that they've got capping enzymes that are encoded in these genomes. Because it's so hard to get from one plant cell to another, almost all of these plant viruses have movement proteins. It's the 3A protein here, but I wouldn't necessarily remember that. Just there is a movement protein. These movement proteins bring the virus RNA to plasmodesmata and help them get into the vascular system of the rest of the plant. Last virus that we talked about in this section was tobacco mosaic virus. It has this wonderful alpha helical structure here, which then binds to the RNA on the inside, gives it this wonderful helically symmetrical structure, and virions which are stable to 40, 50 degrees C. You can dry them out completely, and this was shown by Bierjink in the late 1800s. Already knew that these things were very stable and were replicating inside of apparently living plants. So, any other questions other than what's going to be on the exam? Well, basically everything since the last exam. The point of exams, I think, is not the exam per se. It's the studying that goes into it. Yeah?
I'm trying to remember. Is it the tobacco mosaic virus that has like 6 nucleotides per whatever, and you can like count the number?
Okay. So the question is this the one that I'm trying to get my brain together and say 6 nucleic acids per individual subunit? That's not the one that we've talked about. The one we talked about mostly were the paramyxos and the rhabdos, which had the multiples of 6. Here, it's actually closer to a multiple of 3 per individual subunit. But again, don't worry too much more about that. Good luck on Wednesday.